Hello guys, my topic is blue-green algae, also known as Cyanobacteria. My name is Aryalan Charan, second MSc Microbiology from Sacred College. What is Cyanobacteria? They are photoautotrophs. Cyanobacteria are prokaryote on the basis of size, cell structure, presence of murine, 70S ribosomes and other features. They occur as unicellular, colonical or filamentous organisms. They are mostly gram-negative bacteria in nature. They use water as a hydrogen donor and perform oxic photosynthesis. In cyanobacteria, some are motile by gliding on solid surface. The takes place in flat membranous vesicle under thylakoids. The membranes of thylakoid contain chlorophyll A, beta carotene, oxocarotenoids, and phycobilin proteins. In cyanobacteria, many possess gas vacuoles which are involved. The color of organism is determined by various pigments. Second, blue-green algae. What is blue-green algae? It is widely distributed group of photosynthetic organism of kingdom Cyanobacteria. It can fix atmospheric nitrogen. It can be considered as simple aquatic plants that occur naturally in habits such as rivers, lakes, damp soil, tree trunks, hot springs and snow. Next, methods of mass multiplication. Mass multiplication of blue-green algae classified into four types. They are 1. Trough or tank method. Second, pit method. Third, field method. Fourth, polythene lined method. We see first trough method. This method is basically used in laboratory where zinc and iron trough are used. They are dimensionally 2 into 3 cm in width and 22 cm in height. Trough is filled with about 10 kg of soil and 200 gram of superphosphate is spread on it. Water is poured up to 5 to 10 cm height and calcium carbonate is added to adjust pH around 7. Then started culture is sprinkled over it. Trough is kept in sunlight where blue-green algae developed. In trough method, you can should water it daily. After sufficient growth of blue-green algae, soil is allowed to dry and the dry flakes are collected and packed for algarization. Next, pit method. In this method, under full sunlight, shallow pits are maintained. To avoid percolation, polythene sheets are lined inside the pit. Soil is filled in pit up to 20 cm and water for 10 cm height. Then carbofuran and sawdust are added along with the started culture sprinkling. Then similarly after the growth of blue-green algae, the dry flakes are collected and packed. See, these are some pictures of pit method. Next, field method. This method requires an open and maintained field of about 40 square meter. The plot is watered up to 50 cm height and 20 kg superphosphate is added to it. After maintaining pH around 7, carbofuran is added. Then the started culture is added about 5 kg and is frequently watered. In about 3 to 4 weeks, blue-green algae developed and the soil is allowed to dry. In this method, about 30 kg of blue-green algae can be harvested. Finally, polythene lined method. Prepare the cemented tank, shallow tray of iron sheet or polythene lined pits iron on open area. Width of tanks or pits should not be more than 1.5 cm. Transfer 2 to 3 kg soil and add 100 gram of superphosphate water the pit to about 10 cm height. Mix lime to adjust the pH. Add 2 ml of insecticides to protect the culture from mosquitoes. Mix well and allow to settle down soil particles. When water becomes clear, sprinkle 100 kg of started culture on surface of the water. When temperature remains around 35 to 45 degrees Celsius during summer, optimum growth of cyanobacteria or blue-green algae are achieved. The water level is always maintained about 1 cm during this period. After drying, the algal moss is separated from the soil that forms flakes. It is collected, powdered and packed in polythene bag and supplied to farmers after sealing the packets. The algal flakes can be used as the starter inoculum again. Thank you for watching this video.